Hi everyone, it's a great pleasure to be here. I thought I'd spend the next few minutes just kind of describing a little bit about the problem of antibiotic resistance and how we've been trying to tackle it in the context of bugs that cause problems in CF. And this is work that's been funded predominantly by the CF Trust in the CF Innovation Hub in Cambridge. So I need to tell you that, because I'm going to talk about something that's in a clinical trial, that I've received research funding and consultancy from these two companies, and the one in bold is the one that we're partnering for this clinical trial in a bit. So if you think about so firstly in CF, you can imagine that even with the best modulators in the world, we're still going to be left with a problem for those individuals with damaged lungs and chronic infection with bacteria, that we're going to need to do something about those bacteria. And so it's likely that antibiotic resistance is going to remain a major problem in CF for a while. There's a kind of general problem that there aren't that many antibiotics that target bugs that people with CF get, in particular Pseudomonas. And is that chat?
Yes.
Okay, all right, fine. Sorry, making him. And so it's just worth reflecting on what life was like before antibiotics, and these kind of three examples are kind of conditions where the mortalities were really, really high before antibiotics. And so the kind of worry is that if we don't make any more new antibiotics, we're going to end up in a situation where we run out and we end up in a similar situation to what we were in 100 years ago. So the folks on the left were the people that discovered penicillin, and the chap on the right you may recognise as Darwin, and the moth is the example of how in very quick period of time you can have so much selection pressure that you change animals. And my point really is that there's a kind of inevitability that bugs exposed to antibiotics will develop resistance. And I just wanted to show you how quickly that can occur in an experimental system. And so these scientists in Boston made a kind of tabletop full of agar, which is something that bugs grow on. And they just plated out bugs at either end. And then as you go towards the center, there's more and more antibiotics. And you see that the bugs grow until they reach a point at which there's an increase in antibiotics and they get stuck for a bit. And then one of the bugs gets a mutation and can overcome that level. And very quickly you end up with bugs that are completely resistant to the maximum dose of antibiotics. And my point is that there's kind of an unmet need, an urgent need to address this problem of drug resistance, and that's for CF, but also obviously for other conditions around the world. And there's kind of two ways in which you can create drug-resistant bugs. Drug-resistant pathogenic bugs, pathogenic meaning they cause disease. The first is you start with a pathogenic bug, that's to say a bug that is going to cause a problem. And over time, because it's exposed to antibiotics, you get drug resistance, you end up with a drug-resistant pathogen. And the other way that this can happen is you start off with something that's intrinsically drug resistant, and the example there is abscessus. And then what you do is over time, because of transmission and kind of adaptation to the host, you end up with a bug that is now pathogenic. By either way, you end up with a problem that is a bug that causes disease that you can't kill with antibiotics. And so just to give you this is a kind of example of abscessus, which is it lives in the soil and water and has evolved over millions of years to resist being killed by antibiotics that are naturally occurring in the soil. And that means that they can resist antibiotics in the lung, but it's only partially adapted to the human to begin with. And so for those that are interested, I've listed some publications that you can look at. But what we think is happening for abscessus is that over years, specific clones are selected from the environment. They, because of chronic infection, they adapt within individuals. And if you're in a situation where you can transmit that bug to another person, you can go through multiple rounds of adaptation and you end up with a more pathogenic or nasty bacteria. And what we see with abscessus is what we think happened with tuberculosis about 4,000 years ago. So the big question is, why don't we just make new antibiotics? And the problem with that is that if you imagine the numbers of, if you imagine what normally happens with drug screening is a company has, say, 1 million drugs, and it screens each of them against the bacteria to see if they are effective. But the problem is, if you look at the number of kind of bugs, the number of compounds that you'd need to sample all of chemical space, it's like 10 to the power of 40. That's 10 followed by 40 zeros. And just to give you an example of how massive that number is, there's only 10 to the power of 12 stars in the Milky Way. So this is a huge problem. And so one of the ways in which we can overcome this is a method that's inspired by Lego. So I'm going to talk about that for a second. And the other way is to see how the body kills bugs and see if we can just make that a bit better. And so very briefly in the next few minutes, I'll talk you through the approaches that we've been using at the Innovation Hub to try and overcome this problem. So you can imagine this massive complexity of chemicals that might form antibiotics can be simplified if you break down all chemicals into their building blocks or Lego blocks. And these two individuals here, famous scientists, Tom and Chris, pioneered this, this method of fragment-based drug discovery. And so under these circumstances, instead of looking for chemicals that bind to a particular target, which is what conventional screening is, you just look at the individual Lego blocks, see if they can bind, and then either grow them or merge them to make them into better compounds. And just to show you, I'm going to skip these, but just to show you that this method does work, and we've shown that we can create antibiotics for Mycobacterium abscessus using this method, which is really exciting. The other way, though, is to say, well, hold on a second, let's just put chemicals to one side. How does the body do it when it works, and can we make that better? And if we do that, then one of the things that we found in the lab is that the way that the body kills abscessus is by generating nitric oxide in cells. And so the question we ask is, can we enhance killing by increasing nitric oxide? And so for those of you that are long distance runners or interested in kind of endurance sports, you may have come across this trend now to have beetroot juice. And it's been shown actually in trials that beetroot juice improves exercise and has all these benefits. But it turns out that the chemistry involved when the beetroot juice hits the acid in the stomach, or you can add lemon juice to the beetroot juice, is that it creates nitric oxide in a different way to the body, but creates massive amount of nitric oxide. And so we asked the question whether we could use this chemistry to cause the generation of nitric oxide in the lungs of people with CF who are infected with abscessus. So before we got there, we just took abscessus and grew it up in liquid. And you can see when you add the acidified nitrite in green, we kill all the bugs, absolutely everything. There's nothing survives. And we can take sputum, so phlegm from people with CF, and stick in a little bit of abscessus and then kind of mirror what might happen if we added acidified nitrite through a nebulizer, and we managed to kill all of the abscessus in the samples. And in fact, we can get these macrophages, these cells in the immune system that gobble up bugs, and also the nitric oxide that we produce from the acidified nitrite kills everything. So obviously this is all in the test tube. But what we've also shown is you can take mice and infect them with abscessus. This is work done with Diane Ordway, and then treat them in a similar way that we would do here. And in the dark red is what happens if you treat once a day with the acidified nitrite solution compared to the controls. And just to give you an idea, this is amikacin. This is an antibiotic that's often nebulized. And so it's several orders better than amikacin. So we were kind of so excited by this that we partnered with 30 Tech, which is a biotech company who are funding a clinical trial that Charlie Howarth is running at Papworth Hospital, but also in conjunction with 3 or 4 other hospitals for adults with CF who are infected with abscessus. And hopefully we'll see whether there is an effect or not in 10 people. And if there is an effect, then we can go on and do a bigger trial. But the nice thing is that we're already at a position where we can use this in people. We've got approval for that. It's very safe. to see if it works or not. So just to conclude then, new antibiotics are urgently needed, and the future for existing and future drug-resistant pathogens. The fragment-based approach, the Lego blocks, may provide a simple way to create new antibiotics, and we're doing a lot of work on making that even easier. And then I think there are lots of lessons that we can learn from how the body tries to kill these bugs, and which provides a very simple way, or more simple way, to kill bacteria. And so these are the people that did the work. I'll just leave them there for you to look at if you're interested. But importantly, the funders, the CF Trust, the Wellcome, and the Botner Foundation. And thank you for your attention.
Thank you for such an interesting talk, Andres. I'm just going to put your slides up, Paddy, and then if in the meantime, could you tell everyone what you'll be speaking about?
Sure.
And we're going to save all the questions that I'm sure you have for Andres at the end. So again, if you could put them in the chat, that would be fantastic, or ask them at the end. But over to you.
Great. Thanks, Candice. Hi, my name is Paddy Harrison. I'm going to be talking today about genetic therapies for cystic fibrosis. And I've got slides coming up now. So I'd also like to thank the people who funded the research in our lab in Cork in Ireland, the CF Trust, the CF Foundation, and some money from CF Ireland as well. Find a technical pitch here. There we go. So we know that CF is caused in the lungs when we get this problem. So normally what happens in the lung, we've got a layer of airway surface liquid, and on the surface of the cells we have these hair-like projections or cilia. And on top of that we've got mucus with a few bugs and bits of dirt there. Normally what happens is these cilia beat in a rhythmic fashion, and they'll clear out this material. We can cough this up and spit it out. But occasionally what happens in CF is that this airway surface layer collapses, and then the bugs accumulate, the mucus gets thicker, and this gives rise to the lung disease. So for 60 years, up to about 1989, all that we could really do for CF was to treat the symptoms, try and get rid of the bacteria, try and clear out the mucus. But in 1989, scientists made 4 remarkable discoveries, and I'll just go through those briefly and the impact they've had. First of all, they identified the gene or the genetic material which was associated with this process normally. They identified the messenger RNA molecule which is made from this gene, worked out its sequence and how it encodes a protein which is expressed on the surface of our lung epithelial cells, and that protein is called CFTR. This protein here can be activated, and when it's activated it goes through a conformational change, and these ions, chloride and bicarbonate, are secreted into the airway surface liquid. And this sets up a gradient then for water molecules to leave the cell and increase the airway surface liquid, and then that starts the process of clearance. They also found, the final discovery was the gene sequence which is mutated in cystic fibrosis, the first mutation, and that's shown on the left-hand side there. If you look at that graph initially, it may not make sense, but when you start to align it properly and annotate it properly, this was the discovery of the so-called F508 del mutation, which affects about 80% of people with cystic fibrosis. If you look at the RNA sequence, this is about 4,000 or 5,000 nucleotides, and if you look very carefully, so you can see that white square. If we zoom in with our magnifying glass, we can see that there was 3 nucleotides missing from this genetic information. And when you use this information with the set of, using the same analogy as Andres, Lego bricks, these are all the amino acids you need to make CFTR, and if you follow these instructions, hopefully you get this protein, but with those 3 bases missing, we get this protein and an amino acid left over, the F508, which is deleted. What this means then is that the protein doesn't function properly, the cilia collapse, and the mucus layer gets thicker and the bacteria thrive. Now we know that we've got small molecules, such as ivacaftor shown here, and that can boost the airway surface liquid and bring about big improvements in lung function. But over time, there's a gradual decline in the effectiveness of this small molecule. So what about genetic therapies? Can we fix or replace that messenger RNA or DNA molecule? Well, let's have a look. What we'd like ultimately is to have some vials like this where we could treat individual mutations, or maybe all mutations, possibly with a single dose. To date, the most successful gene therapy trial was performed by the UK CF Gene Therapy Consortium 7 years ago, and the results are shown here in the blue line, which was a mild stabilisation of lung disease. Although it was better than the placebo, you can see here in terms of lung function, it's a long way short of ivacaftor. There's also other therapy trials which are ongoing using the messenger RNA. So this is the wild type sequence. One has been conducted in patients, and in the phase 1 trial, there were small increases in lung function. But with a larger trial, no significant pattern of increase was detected. But there's other companies working on this, and they've had success in preclinical models and are hoping to conduct clinical trials shortly. But in both those cases, that's a transient replacement of the gene or the messenger RNA. So what about mutation-specific options? Well, in this slide here in blue, we've got the DNA in the nucleus, which makes an RNA which travels up to the cytosol of the cell, the main portion, and at the top there is the protein that we're interested in making. So let me just guide you through a few examples. The first one is this catchy name, ACE Transfer Rna. Let me explain how this works in the context of this list of mutations. You'll recognise many of these mutations here. They're listed in order of allele frequency. The ones in red are ones which respond, at least in part, to some of the modulators. But as soon as we move down that list, we'll see several G542X, 1282X, and 553X, which are all stop codon mutations, and none of these respond to the available modulators. These mutations cause a stop in the protein translation process. So this is explained briefly in this slide here. The squiggly line with the blue ball at the top is a transfer RNA with an amino acid on top. And normally what that does will move in, read the RNA sequence, and allow protein synthesis to continue. But if we have a stop codon sequence, just a single letter difference in the nucleic acid code, this transfer RNA can't recognise that sequence and no protein synthesis occurs. But with the ACE transfer RNAs, or anticodon engineered transfer RNAs, the sequence has been changed. They combine specifically to stop codons, and this then allows new protein to be synthesized. And a group in Rochester in New York have shown that this can be used, at least in cell models, for G542X. When you treat them with the ACE, you then get this thick band pretty similar to the wild type. And it's also worked for at least 2 other stop codon mutations, at least in principle. What about some other mutations? Well, if we move down into the nucleus now, in green, I've highlighted a second strategy known as single-stranded oligonucleotides. We'll just skip disease for a second. This is muscular dystrophy, and these are 4 licensed medications which work by skipping exons. You can't really do that in cystic fibrosis in the main, but there's 3 mutations, we recognise the name of at least 2 of those, which affect about 1,500 people worldwide, where this approach could work. And this isn't a new idea. Over 22 years ago, this group showed that by blocking one of these splicing sites, you could get a partial restoration of exon skipping. But what about the clinical application of this? Well, there's been another scientific paper by this group led by Bathsheba Karem in Israel, and they've just announced that they'll be conducting phase 1 clinical trials for 3849 plus 10kb patients with this approach later this year, and W1282X people in early 2023. If we go a bit further, we can move into my research interest, which is gene editing. So this is now trying to fix the DNA sequence directly. And I became interested in this in 2005 when this paper was published in Nature, Genome Editing, Rewriting the Rules for Gene Therapy. And I'll maybe preempt this by answering one of your questions already. When will this reach the clinic? Well, we're not there with CF, but this has already been used in at least one young girl in London to edit T cells. It's been used in several other patients for a rare metabolic disorder. And it's also been used in 2 clinical trials in patients for a retinal disorder, and one I'll just briefly talk about for a liver disorder. So how can we use these gene editing technologies? Well, this particular disease, there's too much of a certain protein rather than not enough, and you can infuse a messenger RNA and a guide RNA to make the editing molecule into a patient over several hours. It then gets targeted to the liver, and when it enters the liver cells, it forms this complex here, which is called Cas9 and a guide RNA. That will then migrate into the nucleus and bind specifically to a DNA sequence and cut that DNA sequence which is causing a problem, and then that will cause a reduction in the gene which is affecting these people. What you see in this graph, the more editing you get, the more of a reduction in the protein you get. And then when this was taken into non-human primates, this worked very successfully, and this has now been tested in 3 people. So the CRISPR gene editing approach has already been successful, at least in 1 clinical trial for 1 disease. But what about CF? That's the problem. If we can insert those 3 base pairs back, we could start to correct some of these CF mutations. Well, let's briefly go back and I'll give you 2 examples. The 3849, we can do this by gene editing as well. The way these graphs work, we've got in black the regions of CF we want and the bit in the middle in white, which we don't want. I'll skip over that graph, but I'll show you how CRISPR can be targeted to bind specific sequences, cut them out, and when they're removed, oops, I think my slide has paused. When they're removed, that will then cause the 2 sections of the DNA to, might be better if you skip out, go back in. That will then cause the 2 sections to join up again, and we get normal splicing. Now, we've used this approach in cystic fibrosis, but this approach has also been used in a clinical trial for a retinal disorder. One of the reasons it was used there was because in your eye, you have a very small target region of the body to attack, but in the lungs, it's a much larger target, and I'll come back to that at the end of my talk. So the final example I want to briefly mention is base editing. In that previous example, we were removing things or destroying things. Can we actually fix them? So with base editing, we've applied this to W1282X, which is the 2nd most common stop codon. The Cas9 protein there in blue recognizes a specific sequence, and it positions this green enzyme into direct contact with the mutation, which in this case is the A residue shown in red. And it can chemically modify that A residue. And then the Cas9 protein is removed. It removes a little bit of DNA with it on the opposite strand, and this can lead to permanent repair of this mutation. So we've shown that this works very well. There's other editing approaches which we can use, which work equally well. But the outstanding challenge is delivery. How will we deliver these Cas9 molecules to the lung of people with CF? And that's one thing that we're now working on in our SRC. Just one idea of the challenge. On the right-hand side here, I've got a molecule of ivacaftor, and on the left-hand side, I've got a base pair of DNA, roughly at the same magnification. But remember, in our cDNA sequences, we have thousands of base pairs of DNA. So if I made the base pairs of DNA the same size as a stamp, the ivacaftor molecule would be the same size as one of the jewels on the crown of the monarch there. And if we think about this in the size of the whole gene, if we put the stamp on an envelope, the whole gene wouldn't be that big. It would be this big. So delivery, getting it into these little bottles and getting into patients is something that we're working on. Okay, I'll finish there. Thanks very much.
Thank you so much, Paddy. Both talks were really interesting. Now we're going to go over to the chat, or if anyone wants to come onto their video and mic live, we're going to take some questions for both Paddy and Andres. I'm going to stop sharing. In the meantime, though, Andres, I was wondering if you could answer one of my questions, sure, which is how long could it take for new antibiotics to be available in clinic?
Okay, so the timelines for antibiotics from the test tube to the clinic is quite long. And we think it's hard to do in less than about 8 to 10 years. What you can do, though, is to develop methods to overcome a lot of the risk at the early phase of developing antibiotics, to make it more attractive for more companies to have a go, more academic groups to have a go. And so hopefully the number of potential targets, the number of potential antibiotics explodes. And then each of them will have either a quicker or a slower route to market, or to clinical trials. it's quite hard to work out if there's ways of shortening that distance between finding a chemical, developing a chemical, and getting it through all the regulations and proving that it works and it's safe. As I hope you kind of appreciated from the second example, though, that if you're able to harness other types of approaches, you can move very quickly into clinical trials. So there's a whole group of ways in which you can kill bacteria by stimulating the immune system. They're called host-directed therapies, and many of those involve human proteins. And to do that, or human compounds, to do that, you can move really quickly. Meaning, in the example I showed you, we found it worked in abscessus 3 years ago, and we're already in clinical trials. And if it's successful, maybe it's another year and a half or 2 years, and it could potentially be licensed. So I think there are other ways in which you can get more rapid wins.
That's really fascinating. Do you think there'll ever come a day where antibiotics could completely eradicate infection from the CF lung?
Well, okay, so I think the CF lung, for those people with CF who are adults now, is a very different place to those people being born today when they're the age of adults. And so I think that there are enormous challenges of getting rid of every single bug in the context of people who have chronic infection. But then again, I think if we have successful antibiotics, then we can reduce the numbers of bugs to the point at which you can hardly detect them. That's kind of almost the same thing. I think that is realistic.
Yeah. And I think we have one question on the chat, which is, what's your view on the use of prophylactic antibiotics? Is this contributing to why we need more antibiotics, and is there an alternative approach?
Right.
That's
okay. So that's a great question. So when we use long-term antibiotics, we're effectively trading two things. We're trading the risk of developing resistance for reducing the bacterial burden for a long period of time and reducing lung inflammation and inflammatory damage. That's the trade-off. And I think there are methods in which we can improve how we use long-term antibiotics to reduce the risk of developing resistance. But unfortunately, there's no way around that. That's the trade-off.
Yeah.
One possible future is to have a set of antibiotics that you can use as long-term prophylaxis and a completely different set for treatment. And therefore, you would never get resistance developing to the antibiotics that you need when you're really ill. Yeah, but we're a way off that.
Okay, that's really interesting. I'm going to move on to a question for Paddy, and that question is, how often would gene editing therapies have to be taken once they're kind of in use in clinics? Do you have an idea of that?
Yeah, so the ideal would be just once. The early data from some of the animal models is potentially just once. It depends how many cells you can correct in the first instance. And this is why this delivery question is foremost in most people's minds working in this field. And the example that I gave in the eye, which is a closed system, the retina, is literally about the size of a ping pong ball. With a skilled surgeon, you can inject gene editing material in there. And there's a few patients now have had this procedure. They're waiting to see what the results are, but you could flood that region of the back of the eye, the retina, and correct those cells. And if that was successful, then that would be a permanent process. In CF, in the lung, it's a much bigger target, first of all. Your lungs are literally the size of a tennis court. So would you be able to get all of the cells in the first instance? And then the lung is also made up of lots of different cell types. At the bottom, we've got the so-called basal cells, which divide and produce all the other cell types. And there's two schools of thought. One is that you would need to target those basal cells, and then once they're edited, they'll then repopulate the lung. But that repopulation process could be quite slow. So another school of thought is if you can target the existing cells and fix them, and they have a long half-life, they could last for months or even years, then that could also be an option there. What we really need to do is to do the experiments in animals to give us an indication, and then follow this up with detailed clinical trials in volunteers.
Yeah. So there's another question on the chat for Andres, but I just want to ask you one follow-on question, which is that I guess the gene editing, if you're talking about kind of similar to the eye example, might be targeted specifically at the lungs. And if that's the case, do you think that there will be a knock-on benefit on kind of other areas of CF, which, for example, the modulators have benefits for digestion and some of the other kind of different non-lung related symptoms? Would you see the gene editing having that same knock-on effect?
Yeah, so that's another great question. So it depends on the route of delivery. If you nebulize it or give it orally, then you will only get the lung. The other option is to try systemic delivery. So in the example I showed where it was infused over several hours, and then it can reach all targets within the body. So we know that that's possible, at least in principle, from animal studies. And then potentially you could target the guts, the pancreas, as we've heard about today, the liver, and so on. And then possibly get at the lung cells from the inside, because the circulatory system has really good access to the lungs, because that's how we get the oxygen into the body to move it around. So potentially that's a route as well, and we're also trying to collaborate with people to explore that.
Yeah, that's fascinating. Thanks for answering that one. So there's a question on the chat from Angela who says to Andres, do you think the approach with regard back to antibiotics will change for those who have been on Orkambi and Kaftrio from day one in terms of antibiotic use?
From day one, when they're, you mean day one of birth?
I think so.
Yeah. So, okay. So, so from the data is better for the people on ivacaftor, because we've had like a decade or so to look at them. But it's likely to be the same for the Kaftrio. Is that once you've got chronic infection, you're probably going to continue having that chronic infection, and for the reasons we were talking about, just getting rid of once it's established. But what's really interesting is the data is now emerging that once you're on these highly effective modulators, your risk of acquiring these chronic infections is reduced. And there's a paper out a few weeks ago showing reduced mycobacterial infections in people with CF on modulators. So kind of extrapolating back then, if you start therapies before there's any lung damage, before there's a dominant bacteria in the lungs, who knows, right? But in principle, you should be able to prevent most of this happening.
Yeah.
The other thing is, do the modulators have an effect on the immune system? Well, they have an effect on the immune system in the sense that less inflammation in the lungs is probably better for the body's immune system anyway. And then, as you may have heard earlier today, we've got some support for this idea that there may be effects of CFTR, the CF protein, on the immune cells, and therefore you might imagine that actually starting these modulators from the very beginning could prevent these infections from ever happening. So a lot of that's speculation, but it's not unreasonable to imagine that we're going to see a kind of complete shift in the bugology of CF.
Bugology. I like that phrase. I might call it that. Okay, so I guess the other thing I was going to ask is now with people who have recently been put on highly effective modulators and their kind of infected, their infection rates and their chronic infected status, have you seen anything? Is there any data to show the modulators have changed that kind of space? Okay,
so I think this is a kind of general consensus view that what we're seeing in the real world use is very similar to what we saw in the trials. So I don't know your experience, but exacerbation rates are dropping to about 30%, right? Number one. Number two, the severity of those exacerbations seems to be dramatically less. And number three, the symptoms experienced by people seems to be very different. And obviously, I'm going to let you comment on that. But from our home monitoring study, we've been able to see how the physiology, because we're asking people to measure SATs and spirometry and activity on a daily basis. We've got very, very granular data, so we can see before and then afterwards. And the interesting thing is the shape of the exacerbation has changed, which is consistent from what a lot of people with CF are saying, that the exacerbations just feel different. And so A, that's fantastic, but B, that's really interesting to understand, well, what are the triggers for antibiotics? Have they changed in some way for when you need? Anyway, so really interesting stuff. We don't know yet.
Okay, thank you for answering that question. There's a question on the chat from Mike, and I guess I'll put this to kind of both of you. It's for an adult who's already got significant colonized lung infection and is not eligible for modulators, is the realistic medium/long-term solution still a double lung transplant? And I don't know if you guys can answer that, but just some thoughts from either of you.
I won't answer the question directly, but I could possibly outline an experiment where you could start to test this. So one of the things with the genetic therapies is, would a genetic therapy lead to the eradication of a chronically infected lung? And one of the problems of trying to do that in an animal model is that we've got some great animal models, but they seem to have a much more severe gut phenotype than humans. And then the lung phenotype is much milder.
Explain what a phenotype is? Sorry to jump in there.
Just the symptoms. So the whole symptoms in the gut is much more severe in these animals.
Yeah.
They're incredibly sick when they're born, whereas most people with CF, even though they have meconium ileus, it's relatively mild and can be managed. Whereas in the lungs, as we know in people, the symptoms there, or phenotype, is very severe and becomes more severe over time. But in the animals, the symptoms tend to be relatively mild. What would be great, and one of the experiments we're trying to design at the moment, is to get rid of the delivery problem by making an animal which would have the CF mutations to start with, and then we can do some genetic tricks to put some drugs in the drinking water, which will then lead to repair throughout the body. You wouldn't be able to do this strategy in humans, but then see what effect that has by simultaneously editing all of the cells in the lung or other organs. And if we had a good model for infection, you could then say, well, we've now fixed every single cell in the lung. What's happened to the infection? And if there was a good enough model, you would actually have a proof of principle answer for, well, actually you do eradicate it, or we still need a lot of extra treatments, physio, antibiotics, and so on. So the jury's out at the moment from the research point of view.
Okay. And Andres, do you have any thoughts around that one?
On the clinical side, so let me fast forward, say, 5 years. What are we going to have then that we're not going to have now that could influence the clinical course of those individuals that aren't eligible or aren't able to have the modulated therapy? And I think you can break that down into two. The first is these new antibiotics, which are already coming through, and they are very exciting. And I think in the next 5 years, we're going to have quite a lot of more antibiotics for Pseudomonas, if Pseudomonas is a thing, and for mycobacteria. So from that perspective, I think that could see a change in how the lung disease develops. And the other, which is probably more exciting because it's a different approach, is the activation of other ion channels in the lung. So there are other chloride channels. CFTR is a chloride channel, like Paddy really nicely described, but there are other chloride channels. And so there are clinical trials at the moment looking at ways in which you can activate these other chloride channels and conversely inhibit the sodium channel, which acts against the chloride channel. And if those work, we should know in the next 2 years, if they work, then it doesn't matter what the genetics are of the CFTR mutation, but you can overcome that by stimulating these other pathways. And if then that would be a game changer, because then effectively you'd be restoring the lung to kind of the level that we're getting now with the modulators. So those are the two areas, if I had to predict, that there'll be game changers in the next 5 years.
Thank you. That's super interesting. Paddy, I have another question for you, which kind of goes back to the gene editing side of things, which is your specialty. Would those treatments that you were speaking about before work on lungs that have a lot of mucus buildup, and would they reach the lung cells?
So again, this is a delivery question. In the CF lung, you get mucus plugging, you have lots of infections, you can even get lung scarring and long-term damage to tissue. One of the issues that people have been looking at is different delivery systems which can penetrate through mucus, and certain virus vectors which have been used are very small. There's a specific size cutoff, and if they're below that, they can actually penetrate the mucus and reach the cells underneath. But this is, again, one of the reasons why we're considering a systemic delivery, because if you have systemic delivery, then that's through the circulatory system. You come out the cells on the other side, and the word you use exactly, you bypass all of that. So both approaches are being considered. There's one other approach which some members of our SRC are also looking at, and that's known as ex vivo gene therapy. So this is to remove a small number of lung cells to correct them in the laboratory, and then to try and transplant them back and get them to engraft into the lung. So again, people are looking at this in animal models. Yeah. So that's been used for, again, some other disorders with bone marrow diseases. We're looking to see if you could possibly do that in the lung.
And if in theory it works that they go back into the lung,
yeah,
how does that affect the rest of the lung? Does that kind of spread in some...
Again, really interesting questions, and we can speculate, but we don't have the full answer.
Yeah.
One idea is that there's a growth advantage for wild type cells or corrected cells relative to CF. So it's slightly different from the graph that Andres showed where the bacteria are growing. If you've got lungs in the implanted cells in the lung and the corrected ones grow faster, then they'll outcompete these, and you may start to get patches of the lung which are doing slightly better.
Yeah.
Again, that's just speculation at the moment, but hopefully that's the case.
Yeah, okay. I'm going to check if there's any more questions in the chat, and I think there's one from Jennifer, which is how long do you think it will be before gene editing, I guess, is in practice in the clinic and people are able to start taking gene therapy medicines and
yeah, so there's I can answer that in two ways. So with gene therapy, which was the idea of adding the gene back or the cDNA back, this idea came out in the 1970s, and the first licensed medication has only been around for 3 or 4 years with this, again, for a retinal disorder. Yeah. With gene editing, the first paper I saw was 2005. And by 2015, so less than 10 years, it was being used in an experimental technique for a rare form of cancer. There's now 2 clinical trials using CRISPR. There's another clinical trial which has been completed using another gene editing technique. So what I'm really excited about is there's a few other diseases which have a bit more preclinical data than us. We can do the editing now. I think we need to work on the delivery and get some nice data in animal models showing the efficacy. And then some of the talks this morning I think was interesting in the antibiotic arena, trying to get a pipeline ready for companies and academic groups to feed in. I think we need to do the same with gene editing and have people lined up so that when this is a bit more advanced in a few years' time, people then have the confidence and the ability to take this through to clinical trials as well. So you can never put a number on these things. Yeah. People always say 5 to 10 years, but I don't think it's unrealistic that within 5 to 10 years you'll see at least some people undergoing this as an experimental investigation.
Well, thank you for explaining. I think it's also so important that people start to understand what these trials could look like, so that when they come around, you have the confidence to know whether that's something you'd want to be a part of. And thank you so much for letting us know more about antibiotics. There's one final question, which we just about have time for, from Catherine, which again, I think we'll put to both of you, but maybe Paddy, you'll be a bit better to answer this one. What do you envisage is the end game? A patient-tailored combination of modulators and gene therapy, or one or the other? And I guess how do antibiotics play into that as well in the end game?
Either or something completely different.
Yeah.
It really doesn't matter what it is if it works.
Yeah.
I believe that. I think the research we're doing is very important and interesting. I think what Andres is doing is very important and interesting. There's loads of stuff we've not heard about from other research groups who are targeting different genes, different strategies, different small molecules. Yeah. Whatever works for people as soon as possible, as safe as possible.
Yeah. I agree. I don't know, Andres, do you have anything to add?
Oh, no, I totally agree. Any port in a storm.
Yeah,
right?
Exactly. Yeah. Great. Well, thank you so much, both of you. I think I can say that's been absolutely fascinating, and I hope everyone online has agreed with that. And so that's the end of our session for Breakout B. A big thank you again. The rest of the conference will be streamed on the main kind of platform from 3 p.m. And the next session, which Catherine will introduce, is Research for Tomorrow. So we will hopefully see as many of you there as we'd like to be there. So thank you again for joining.